This is a review for the unit test, all about cellular processes. The unit test is going to cover standards 2.5, 2.6, 4.3, 4.5, and 2.6. The first thing that we need to talk about is ATP. ATP, energy. That's right. ATP is an energy molecule. ATP stands for adenosine triphosphate. Some people get confused because although the A is separate, the T and the P are part of one word. But this word is important because you can see what is inside of ATP. ATP is made of an adenine, hence the word adenosine. Tri, think of a tricycle, means 3. And right next to it says 3 phosphates. This is the most important part of an ATP molecule, these 3 phosphates. There's also a ribose made that is in ATP. Remember, if something ends in OSE, that means it's a sugar. So let's take a closer look at why ATP is so important and where the energy is stored. All of the energy in ATP is stored not here, not here, not in adenine, not in ribose. The only place where energy is stored is in this bond between the second and third phosphate. Remember, a bond is what holds molecules together. So all of the energy is right here. What that means is when the bond is broken and it's no longer held together, energy is going to be released. And then you're going to have a molecule that's known as ADP. D stands for di, and di means 2. That means that there are now only going to be 2 phosphates as opposed to 3 because this bond was broken, and you would just have this molecule right here. Exactly the same except just missing 1 phosphate. But luckily, we can even store energy in ATP. All we have to do is add this last phosphate back on and restore this bond. And now all of the energy is back because remember, all of the energy is held right here. And now we would have 3 phosphates, so we would have a molecule of ATP being made. So that's how ATP works. What we're going to be talking about is how ATP is made in cellular respiration. But before we can use ATP, we have to make that through food, through sugar. And the process that plants and some algae use to make their own food is called photosynthesis. Photosynthesis takes place in the chloroplasts of plants. Photosynthesis only happens in autotrophs. Those are organisms that can make their own food. Think of the word auto as self. They can make food themselves. So they can make their own food. You have to know that term, and you have to know that this takes place inside of the chloroplasts. Cellular respiration is what is now going to take that food and it's going to make it into ATP. This takes place in the mitochondria, and this is what we do because we are considered heterotrophs. We cannot make our own food. So we're going to dive into these 2 processes in more detail. The first one is photosynthesis. You need to know the equation for photosynthesis, which means what it uses and what it makes. There are 3 things that photosynthesis uses: CO2, which is carbon dioxide, H2O, which is water, and energy. They get this energy from the sunlight. They use all 3 of these things and they make 2 things. They make glucose, which is sugar, and O2, which is oxygen. Remember the song. You use the light and the water and the... You use the light and the water and the... You use the light and the water and the... to make sugar and O2. However you need to remember it, you need to know it. So these are what is used in photosynthesis, and this is what is made. We give fancy terms to the things that are used and the things that are made. The things that are used are called reactants. They're the ones that are reacting. And the things that you make are called products. That's easy to remember. Think of a final product in any of your classes or shops is the final thing that you make. So you might hear, instead of what does photosynthesis use, what are the reactants of photosynthesis, and also for products as well. We can represent that in a picture form. This is one thing that you might consider drawing on your unit test to help you remember what is used and what is made in photosynthesis. Photosynthesis happens inside of chloroplasts. We don't have chloroplasts because chloroplasts contain a pigment that makes plants green. That pigment, chlorophyll, is located inside of these things that look like coins. Each coin is called a thylakoid, and a whole stack of coins is called a granum. Inside of the chloroplast, there's also liquid. Just like there's cytoplasm in our cells, there's a type of cytoplasm that's found inside of the chloroplast, and that is called stroma. Think of straw. You suck up liquid with a straw. So stroma is the liquid portion. There's two main parts of photosynthesis. There's the light-dependent reactions and the light-independent reactions. If it's dependent, it means it needs the light. If it's independent, it means it doesn't need the light. These happen in two different places. When you need the light, it's going to take place in the thylakoids, because remember, that's the part that contains chlorophyll, that pigment that absorbs the energy from light and makes a plant green. The place that does not need light is called the stroma, that liquid portion. You can also hear the light-independent reaction referred to as the Calvin cycle. The guy who found this out named it after himself. Lucky him. Cellular respiration is the exact opposite. We just made glucose in photosynthesis, and now in cellular respiration, we're going to be breaking that glucose down to make it into a form that our cells can use, which is ATP. This occurs in the cytoplasm, but mostly inside of the mitochondria. A lot of cells perform cellular respiration, but you're going to see the more energy that a cell needs, the more mitochondria the cell has. It needs to make more energy. For example, our muscles and nerve cells are constantly using energy to help us move and coordinate our body. So they're going to have much more mitochondria than, say, the skin, which doesn't need that much energy. Again, you need to know the general formula for photosynthesis and for cellular respiration. It's easy. If you know one, then you know the other. So we just made this in photosynthesis, oxygen and glucose, and now we're going to be using oxygen and glucose. It also makes sense. If cellular respiration is what we are doing, we're breathing in oxygen and we're eating sugar. And we make three things with glucose and oxygen. We make two wastes, carbon dioxide and water, which we breathe out, and we make energy in the form of ATP. So really, cellular respiration is all about taking the energy of sugar and changing it into a form that our cells can use, which is ATP. Cellular respiration happens in a number of steps. You really just need to know the order of the steps. Glycolysis is the first step, and this takes place in the cytoplasm, the liquid portion of the cell. Then comes the Krebs cycle, and then the electron transport chain. This is really where most of the ATP is being made. An easy way to remember the order of these processes is glycolysis is one word, Krebs cycle is 2 words, and electron transport chain is 3 words. So remember, 1, 2, 3. Cellular respiration needs oxygen, but if no oxygen is available, something different is going to happen inside of cells. If there's no oxygen available, it's called anaerobic respiration. The AN before aerobic means that it's the opposite of aerobic. It does not use oxygen. Sometimes you also hear that referred to as fermentation. There are 2 types of fermentation or anaerobic respiration. The first is alcoholic. This is what happens in yeast. Yeast are responsible for making our alcohol and a little bit of carbon dioxide, which makes your alcohol bubbly. Think of beer. And the second type of fermentation is called lactic acid. This is what's happening inside of our muscle cells. This is what happens when you first start to work out. You don't have enough oxygen available, and you get that burning sensation in your muscles because lactic acid is being made. This doesn't make too much energy, only 2 ATP, whereas in cellular respiration, 36 ATP are made. So it's much better to have oxygen than to not have oxygen. So to summarize photosynthesis and cellular respiration, we're going to use some of these fancy terms that we went over, products and reactants. Basically, what you need to know about these two is that what one process makes, the other uses. So let's look at that. Photosynthesis makes glucose and oxygen, and that is what cellular respiration uses. So again, what one makes, the other uses. And vice versa for cellular respiration. Cellular respiration makes ATP, but it also makes these two waste products, carbon dioxide and water, which we breathe out. And photosynthesis is then going to take and use those products in addition to light to make glucose and oxygen. So we say that cellular respiration and photosynthesis occur in a cycle. Energy is being cycled, waste products are being cycled as well. Now, how do we get that oxygen in and let carbon dioxide out? That's the job of the respiratory system. There's a lot of different parts, and you need to know the order of which air passes through. It's going to come into your nose, through your nasal cavities, or in through your mouth, and it's going to go into your throat. The throat is not shown here, but your throat is also called your pharynx. If it's in your throat, it needs to decide which way to go. If it's food, it wants to go down the esophagus to the stomach. If it's air, it's going to pass through the larynx, which is a fancy word for your voice box. It's going to go down the trachea, branch into 2 bronchi, which go into the lungs. Then it's going to branch even further and further, which are called bronchioles. And at the very end of the bronchioles, you're going to see these tiny little air sacs, which are called alveoli. Here's a zoomed in picture of the air sacs, or alveoli, and you can see that they're surrounded by these tiny, tiny blood vessels. Those are called capillaries. This is going to become important later on. The other thing to note is that there is a thin muscle right below the lungs called the diaphragm. This is going to help you to get air in and out. So let's take a closer look at each of these parts. Here we have the parts in order. The first part is the nose. The job of the nose is to filter, warm, and moisten the air that's coming in. It's better to breathe through your nose than your mouth because your mouth cannot filter the air like your nose can with the tiny little hairs that are in your nose. The next part is your pharynx. Your pharynx is a fancy word for your throat. This is where food and air pass. Food and air need to decide which way to go. What helps them decide is the epiglottis. This is a flap of tissue that covers your windpipe when you're eating. And when you're breathing, that epiglottis is going to stay open so that air can go down to your larynx and your trachea. So this is a very important part to help you from not choking, to not choke. Then comes your voice box, your larynx. An easy way to remember that your larynx is your voice box is think la 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 la. That is producing your sound or your voice. Then comes your trachea. Your trachea, it can also be known as your windpipe because this is where wind or your air is going down. Your trachea then branches out into 2 bronchi. Those are going to lead into the 2 lungs. Once inside of the lungs, those bronchi are going to branch into smaller and smaller branches, which are called bronchioles. It's almost the same exact word, but all we added was O-L-E-S, which means small. At the very end of these bronchioles are those air sacs, which are again called alveoli. Those alveoli are covered in tiny blood vessels called capillaries. What's really important is this is the smallest part and in contact with the blood. So this is where gas exchange takes place. This is where gas is going into the blood and gas is going out of the blood. So which one is going in and which one is going out? Let's take a look at that. Oxygen is going to go from the alveoli into the blood because we need oxygen in our blood for cellular respiration. CO2 is going to move the opposite way, from the blood into the alveoli so that we can breathe it out. This all happens because of diffusion, where molecules move from a high concentration to a low concentration, from where there's more to where there's a little bit. The diaphragm is that thin muscle right below your lungs, and this is what's helping you to get air in and out. When you're inhaling, your diaphragm is going to move down, and it's going to allow for the lungs to expand and have as much room as they possibly can. When you're exhaling or breathing out, your diaphragm is going to contract and move up, which is going to cause the lungs to be smaller and push the air out of your nose and mouth. The last thing that we're going to talk about is our 3 types of muscles. There are 3 types of muscles: skeletal, cardiac, and smooth. Skeletal muscle is found attached to your bones, hence the word skeletal. It's attached to your skeleton. This is considered voluntary, and it's the only type of muscle that is voluntary. That means that you control it. So if I wanted to slap someone across the face, that's using my skeletal muscles. I control it. Cardiac is found in the heart, and this is considered involuntary. You do not control your heartbeat. It happens on its own. And smooth muscle. Smooth muscle is found in the organs like your stomach and most of your digestive tract. This is also involuntary because you are not controlling the passage of food through your digestive system or any other organs. A little bit more about the skeleton. The skeleton is made of 206 bones. It's divided into 2 parts. The axial skeleton, which is colored in red. This is what helps to support the body, provide balance, and protection of internal organs. It's made up of 3 parts: the spinal column, which is your spine, the skull, and your rib cage. The second part of the skeleton is called the appendicular skeleton. Think of appendages, what's on the side. The side is your arms and legs. This is what allows for gross movement, which means walking and lifting and things like that. The structure of bones are very important. The outside layer is made of compact bone. This is hard and dense. This is the site where minerals are stored, such as calcium. The whole bone isn't made of compact bone, but rather most of it is actually made of spongy bone. This is because this is more lightweight, but yet still strong. Spongy bone isn't soft like a sponge, but it has holes like a sponge. This makes the inner layer of the bone. Inside of the bone is also bone marrow. There are 2 types of bone marrow, red and yellow. Red bone marrow is where many blood cells are made. That's easy to remember because red marrow makes blood cells. Blood is also red in color. And yellow bone marrow stores fat. Fat is yellow in color. Some other important terms are joint. Joint is the place where 2 bones meet. A joint is held together by ligaments, so ligaments connect bone to bone. Another thing is cartilage. Cartilage is found in your ears, the tips of your nose, and your ribs, as well as many other places such as the ends of bones. The thing that's different about cartilage and bone is that cartilage is strong like bone, but it's flexible. It can easily be bent and not broken. The reason why we're learning muscles and the skeletal system together is because you need both muscles and bones in order to move. Muscles are connected to your bones by tendons, so tendons connect muscles to bones, whereas ligaments connect bone to bone. How this coordinates movement is that your muscles pull on bones when your muscles contract. This causes your bone to move. Muscles also work in pairs. When one contracts, the opposing one relaxes. The last part of your unit test will include the cell cycle, which is including everything from G1 all the way to mitosis. We learned that the cell cycle occurs in 4 different phases: G1, S, G2, and N. Briefly, let's talk about what happens in each stage. G stands for growth. So the first time the cell grows, it's going to grow to a mature size and just do its normal metabolic functions, making proteins, making organelles, etc. The S stands for synthesis. What's being made in the synthesis phase is a copy of DNA. The DNA has to be copied before a cell divides so that each cell has the same amount of DNA in each cell. G2. G2 also stands for growth, but now we're growing a second time. The cell is going to double in size and prepare for division. That means our organelles are going to be doubled. And the last phase is M for mitosis, which we're going to talk about in more detail. One thing to realize is that DNA can be in two different forms. The first form is chromatin. That is when DNA looks like string. This is when a cell is not dividing. So this can be found during the G1 phase as well as G2 and S. During prophase, that chromatin condenses into what are called chromosomes. Chromosomes are made of two identical sister chromatids when they're replicated. So one of these is a sister chromatid and one of these is a sister chromatid. They are attached to each other by what's called a centromere. This is represented by this yellow dot right here. This is holding the two together, and these two sister chromatids are identical. Now, mitosis is technically just the division of nuclei, and then we'll see how the actual cell is split in half. Mitosis is going to come directly after G2, although not all cells go through mitosis. Mitosis occurs in 4 stages: prophase, metaphase, anaphase, and telophase. An easy way to remember is it spells PMAT, or Pablo made amazing tacos. You need to know what happens in each of these phases. In the first phase, prophase, which is the longest phase of mitosis, the most important thing that happens are chromosomes become visible. That means they go from looking like string, which is chromatin, into looking like chromosomes. So now we can finally see the chromosomes. Another thing that happens is the nucleolus disappears. The nucleolus is the smaller ball inside of the nucleus. The nuclear envelope also breaks down. What that is that's the outside layer of the nucleus. So that is going to break down so that the DNA can easily split in half. So here we have a picture of a cell in interphase. Again, you can see that the chromosomes are not visible, and here they are becoming visible. The nuclear envelope is breaking down, and the nucleolus has disappeared. Some other things are happening in this picture, though. One thing is that these centrioles are going to start separating. Here they were together, but now they're going to be going on opposite sides of the cell and forming a spindle. The spindle are these fibers right here. We're going to see why they become important in a little bit. Something to mention is that plants do not have centrioles, which are these right here. Instead, they just have the centrosome, this area, so they're going to use that to organize their spindle. These spindle fibers are later going to attach to these sister chromatids, and they're going to be moving the chromosomes around the cell. The spindle fibers are going to attach to the sister chromatids right in the middle of the centromere. In metaphase, an easy way to remember what happens is M for metaphase, M for middle. The chromosomes are going to line up in the middle of a cell like you see right here. This is obviously the second phase, PMAT. The spindle fibers are now attached to the centromeres, and they're going to be pulling the chromosomes so that they are lined up again in the middle. In anaphase, easy way to remember is that A for away. Now what's going to be happening are the chromatids are going to be pulling away from each other. They're going to be separating and becoming individual chromosomes again. The spindle fibers are responsible for moving these chromatids to opposite sides of the cell. They don't just magically move where they need to go. They have to be pulled by those spindle fibers. And the final stage, telophase. Think T for telophase, T for 2. This is the last phase of mitosis, and this is where everything that happens in prophase is undone. Chromosomes are now going to unravel and loosen into their string-like form called chromatin. The nuclear envelope is now going to reform around each cluster of the chromosomes. Remember, now we have 2 being formed. The spindle is going to be broken apart, and you'll no longer see spindle fibers. The nucleolus is now going to become visible in each dot or nucleus. So it's the complete opposite. So here we have each of the steps. In interphase, again, you cannot see the chromosomes. They become visible in prophase, as well as the nuclear envelope breaking down, and there's no nucleolus present. the centrioles start to move to opposite sides and form these spindle fibers. In metaphase, they line up in the middle of the cell. Again, these spindles are attached to the centromere of each sister chromatid. Then in anaphase, they're moved away from each other so that each side of the cell will have an equal number of chromosomes. And in telophase, everything that was done here is going to be undone. So now we're going to see the uncoiling of chromosomes so that chromatin is formed. We no longer see a spindle fiber like we do here. Our nucleolus is going to reappear in both nuclei, and the nuclear envelope is going to surround the DNA so that there are two nucleuses. And the final step that we're going to talk about is cytokinesis, which is actually dividing the cell into two. Cytokinesis is the division of cytoplasm. It's dividing the cytoplasm of the cell so that there are two equal cells. This is technically happening at the same time as telophase, although it's a separate part from mitosis. So the membrane is basically going to pinch until the cytoplasm is separated into two equal parts. Each side will have the same amount, but it's a little bit different in plants. We'll see that on the next slide. So each cell would have its own nucleus, but identical, with 46 chromosomes in our cells, and it would also have all the cell parts that are not here. So each one would have a mitochondria and vacuoles and Golgi apparatus, etc. So in the end, we call those two cells identical daughter cells. In a plant, plants have cell walls, so it's a little bit more difficult. Instead, a cell plate will form in the middle, which is eventually going to form the cell wall. So last but not least, why do we even need mitosis? We're learning about all the steps, but why is it important? One reason is that some protists, and fungi are going to be using mitosis for asexual reproduction. They're just going to be dividing in half, and that will be the new organism. It's also responsible for forming somatic cells. Somatic cells are a fancy word for our body cells. That means our skin cells, our muscle cells, hair, etc. In order to create new ones, we have to go through mitosis. Now, in bacteria, it's a little bit different. Bacteria are considered prokaryotes. Remember, pro rhymes with no. These organisms have no nucleus, so they can't go through mitosis because mitosis is the division of nuclei. So they go through a process called binary fission. It's very similar to mitosis. The DNA copies itself just like the S phase. The DNA is going to separate just like anaphase, and the cell splits in two identical pieces. So the process is similar in that there's going to be two identical cells forming, but this process is very fast and very simple because not only is there no nucleus, but bacteria don't have any organelles as well. They don't need to copy their mitochondria and Golgi apparatus and endoplasmic reticulum because they don't have any. The only thing that they're going to be copying is their DNA. This means that bacteria can divide about every 20 minutes. That's extremely fast. Much faster than the whole cell cycle that our cells go through.